Okay. Welcome to What's New in MPMI. And I'd like to first, well, let me introduce you to our top 10 series, because that's what this is one of the next, I don't know, installments of. So some time ago, when we all gathered in person at the IS-MPMI conference in Scotland, we spent a long time brainstorming with the community and also the larger community who could not be at the meeting about what are the unanswered questions that face us today in our field of molecular plant microbe interactions. What are the things we don't know? What are we striving to understand? And one of the really important questions that floated to the top was this question of how does virulence evolve? And so that's what we're going to be focusing on today with Soledad Sacristán as our speaker. But first I wanted to introduce Tessa Birch-Smith, who is Associate Editor-in-Chief of MPMI and is my co-host today. And yes, and I'd like Tessa to give you a heads up about our next focus issue, because I imagine there will be many people here who will be interested in contributing.
Right. So our next focus issue, if Ashley could share that with you, our next focus issue will be, well, it's a 2023 focus issue, but the title is The Plant Endomembrane System in Molecular Plant Microbe Interactions. And the deadline for submission is June 30th, 2022. So you still have several months to get a manuscript ready. We have a great lineup of guest editors. They include Aiming Wong from Agri-Food Canada, Dong Wong from UMass Amherst, and Dr. Yoon Suk Park from the University of Wyoming. So if you work in this area at all, we encourage you to submit a full research article or a review, or even a short article, short communication. So there are many benefits to participating in this focus issue, including, of course, increased readership because you are publishing among your peers. So please consider this special issue as you prepare your manuscripts this coming year.
Thank you. Yes, I'm really looking forward to this one. I think there's a lot of exciting work going on, and I hope that we will be able to include some of your work in MPMI.
Yes.
Okay, I think it's time for our main event. I'd like to turn it over to Soledad Sacristán, who will introduce herself. And why don't you share your screen now and give our seminar. And I just want to remind you that to please type your questions for Soledad into the Q&A box at the bottom, and we will read the questions at the end and try to answer as many of them as we can. Okay, Soledad, thank you so much for coming to contribute to MPMI.
Oh, thank you. It's a pleasure to be here, and thank you for the invitation. So I'm Soledad Sacristán. I'm plant pathologist. I teach at agronomy school at Universidad Politécnica de Madrid, and I do my research at Centro de Biotecnología y Genómica de Plantas. And I'm here because I have written this review together, and it has been a pleasure to write it together with very nice plant pathologist, Erika Goss from Florida, University of Florida, and Sebastian Ives van den Akker from the University of Cambridge. And well, just a little bit of my background. I did my PhD on virus evolution, and then I did a postdoc on evolution of virulence in powdery mildews. So the evolution of virulence activities in plant pathogens has been really central in my career. And well, I think that currently in these days, the evolution of virulence of pathogens interests to everybody. We have sadly having very interesting times for people interested on virus evolution. And maybe, well, I think that we now have the feeling that in the war against pathogen evolution, we are far from winning the war. And we can be happy if we manage to win some battles, or at least to anticipate them, to make the losses not so worse, not so bad. So for that, we really need to know what's going on. So this is what is this review about. At the beginning, when we were proposed, the question formulated as how do pathogens evolve novel virulence activities, we found it a bit strange. We were more used to questions such as the evolution of virulence or the evolution of pathogenesis. I will explain these two terms later. But after thinking about it, we thought that it was fine, because this formulation, how do pathogens evolve novel virulence activities, was very inclusive. And yes, we wanted to do a very inclusive review, not going much in depth, because there are many other reviews that go into the details in the various points that we addressed, but touching as many aspects and pathogens as possible. I have tried to condensate this work in 30-40 minutes. I hope I manage. And I have mostly tried to bring different examples. So I will show the work from many other scientists rather than my own work. And I thank all those people for their work. So how do pathogens evolve novel virulence activities? For asking this question, or for answering this question, maybe we need to know first what are virulence activities, or even what is virulence, and how does evolution work. So what is virulence? There has been confusion around the term virulence among plant pathologists since Van der Planck, which is a very famous plant pathologist, used it to describe the capacity of a pathogen to infect a particular host genotype, so host-resistant genotype. So we are used to say this strain or this isolate is virulent or adherent to that resistant host. But in other fields, such as animal pathology, virulence is usually related to the degree of harm that infection causes to the host. To solve this confusion, the American Phytopathological Society adopted the conventions of using pathogenicity as a qualitative description of the ability of a pathogen to cause disease, and virulence as a quantitative measure of the degree of disease, of the degree of pathogenicity. I have borrowed this picture from a work of Matthew Moscow and collaborators about rust resistance in barley. It is about resistance, but I think it illustrates very well how the pathogens can have different degrees of cause different disease or not, or different degrees of disease. So for including both pathogenicity and virulence and other pathogen characteristics, we decided to use the term virulence activities. And we have considered virulence activities as anything that enables pathogens to infect and multiply. So increased pathogen fitness, with the consequence of causing disease in either the qualitative or pathogenicity or quantitative virulence senses. Therefore, novel virulence activities include the adaptation of pathogens to infect and multiply in the host, to transmit to other hosts, and to survive in the environment. So adaptations to host immunity and physiology, host species, genotypes or tissues, the environment, the production of more efficient reproductive structures, interaction with vectors, are competing or cooperating better with other microorganisms in the ecosystem. So these novel virulence activities may evolve by acquisition or adaptation of virulence factors, which are genes or gene products that contribute to the ability of a pathogen to infect and multiply in the host, and virulence-related factors that are gene or gene products indirectly involved in virulence activities. And how does evolution work? Well, evolution is something complex. There are many concepts, many factors involved. There are many works about different aspects of evolution. So I cannot really explain all about evolution here. So for simplicity, I'm just going to address the concept of natural selection, the evolution of natural selection, where the evolution of an adaptive trait, such as virulence activities, is based on the selection exerted on variants that have been generated de novo or arrived from elsewhere. So we have many variants initially, and selective factors post-selective pressure that selects for some variants in particular. In this case, those that have higher fitness. So pathogen fitness relies on the ability to, so here I will address first the selective factors, virulence activities, and then the genetic mechanisms, some genetic mechanisms that have drive this variability in the pathogen population. So pathogen fitness relies on the ability to multiply in a host and to transmit to other hosts. So host is the main driver for the evolution of virulence activities. However, besides the host, environmental factors during pathogen life cycle may influence the direction of evolution, so lead to new virulence activities. Host has aspects that can drive the evolution of novel virulence activities are host pathogen coevolution, the infection cycle and its trade-offs, and the adaptation to new hosts. So we all know that plants impose physical and biological barriers that pathogens must breach to infect and multiply. For example, plants recognize microbe-associated molecular patterns or pathogen-associated molecular patterns and trigger immune responses. In the widely accepted zigzag model of plant pathogen interaction developed by Jones and Dangle, pathogens overcome these immune responses by the use of effectors, which are secreted proteins or other molecules that contribute to pathogen fitness in the host. Plant-resistant R gene may recognize an effector, termed then virulence genes, and trigger further immune responses. In such a case, the host is selecting for pathogen alleles that avoid this response, including loss of function. Loss of function mutations in AVR genes refers to alleles that evade immune receptors, although they may maintain in whole or in part their virulence activity. Novel virulence activity can also be gain of function events. For example, another effector that subverts this response, immunity response, or alters plant physiology in some way that benefits the pathogen. And plants in turn can evolve novel resistant gene specificities in a coevolutionary process. The result of coevolution by R genes that recognize AVR genes is described by the gene-for-gene model, in which dominant resistance will happen when there is this recognition, and in other cases, when R genes or AVR genes are not present, disease is produced. Here I bring an example of this gene-for-gene interaction in the system Cymoseptoria TTC and wheat, although there are many others. In the system, wheat varieties that carry STB6 gene are resistant to pathogen strains that have AVR STB6. But these strains are pathogenic to wheat varieties that do not carry STB6 gene. Pathogen strains that are not recognized by STB6 gene cause disease in both wheat varieties. But we can see that 1E4 isolate, virulent for STB16, causes more disease and pycnidia on the susceptible wheat variety compared with the other isolate 1A5, which is resistance against STB6. So there is a so-called cost of virulence for this isolate 1A5 in absence of resistance. And the result in the pathogen population may actually depend on the extent of this cost of losing the effector. If the cost is low, the variants or the effectors will be lost. If the cost is higher, the effector will not be lost, and variants with the AVR gene will remain in the population. And we will see a diversifying selection. Another option is not losing the effector, but modify it. And we can see signatures of this diversifying selection in these modified pathogen variants. Continuing with the example, with the previous example of Song and collaborators in Cymoseptoria tritici, Brunner and McDonald analyzed 30 strains of Cymoseptoria tritici and could identify sites that showed diversifying selection, comparing virulent and avirulent strains. So, but virulence factors function by manipulating plant biology to allow or improve pathogen growth and transmission. For that, they have targets in the host. In this case, host resistance may be attained by recessive alleles of the host target that are not recognized by the pathogen virulence factors. But pathogens, in turn, may evolve by modifying its virulence factors so they can again interact with the modified host targets. In this case, host pathogen coevolution follows the matching allele or inverted gene for gene model. Here, disease is only produced when the pathogen virulence factor matches the susceptibility target of the host. And in the other cases, we have resistance. There are several examples of this type of interaction, such as the S genes as targets of effectors in hemi and biotrophic fungi, host-specific secreted toxins triggering R gene-mediated cell death in some necrotrophic pathogens, and host eukaryotic translation initiation factors and VPGs proteins in potiviruses and somavirus. I have brought here an example of this last case. In the case of rice yellow mottle pseudomovirus, which needs to bind to this rice translation initiation factor for a compatible interaction. Variant RYMV12 of this rice translation initiation factor carries non-synonymous mutations that interfere with direct interaction with the direct interaction with the VPG, causing a resistant phenotype. However, there are pathogenic viral strains polymorphic in a particular site of VPG, and these strains do not bind to the original rice translation initiation factor and cannot multiply in this host, but do bind the RYMV12 form of the host protein, and therefore these viral strains will prevail only under the selective pressure of the deployment of RYMV12 gene resistance gene, which is recessive. So we know some things about how pathogens can co-evolve with plant hosts, and we have some models that describe this coevolution in terms of recognition of matching between pathogen and host factors. However, in fact, we can say that most of our knowledge is biased to understanding effector evolution to evade host immunity, but we still have much to learn about effector adaptation to existing host factors. This is related with the fact that the precise function of effectors and their host targets are still unknown in many cases. Also, effectors are not just secreted proteins, there are other non-proteinaceous molecules with effector functions, and it is not so clear if these encompassed by existing evolutionary models. It is generally assumed that there is a cost of virulence activities in the absence of selection that maintains the polymorphisms in the population, but in many cases the costs are not found. So the impact of the costs and how to measure them are not yet fully resolved. And gene-for-gene and matching allele interactions are relatively simple, but increasing evidence points to interactions being more complex. We can realize about the complexity if, besides the initial steps for the establishment of plant-pathogen interaction, we look at the whole pathogen cycle. As I said at the beginning, virulence activities are not just directed to infect the host, but also to colonize its tissues and transmit to other hosts. The process of pathogen multiplication in the host is complex, and the host might exert selection for different pathogen alleles or combinations of alleles at various stages of infection, growth, replication, and transmission. So the complex relationships among traits affecting different stages in the infection cycle may result in trade-offs between virulence activities. And these trade-offs can vary among pathogen genotypes and ecological or host context. One of the most generally assumed of such trade-offs is that between pathogen virulence and transmission. Pathogens may need to attain a within-host multiplication level that allows successful transmission, but abundant multiplication can cause so much host harm that it limits available time for transmission. Therefore, models of virulence evolution predict an optimal virulence level that maximizes pathogen growth and transmission. So the trade-offs in virulence activities within and outside the host may result in virulence in the sense of host harm not being maximized when pathogen fitness is maximized. In an extreme case, virulence is reduced in pathogens that are vertically transmitted through the seed to allow the plant to complete their reproductive cycle. In this work of Pagan and collaborators, cucumber mosaic virus virulence was significantly reduced after 5 generations of vertical transmission through seeds in a serial passage experiment in Arabidopsis thaliana. At the same time, the ability to transmit by seed was increased, and this didn't happen with those strains that were passaged through was transmitted horizontally, the same passages but horizontally by mechanical transmission. Adaptation to different hosts may also impose trade-offs in the evolution of virulence activities. It is assumed that the ability of generalist pathogens to explode multiple different hosts entails costs, so generalist pathogens will have lower fitness than either specialists in its preferred host. However, the ecology of some pathogens may force them to change host during their life cycle, selecting for generalist adaptation despite the costs. There can be different causes to these trade-offs. For example, antagonistic pleiotropy, yes, which means that mutations that are beneficial in one host may be deleterious in an alternative one. A second mechanism can be mutation accumulation, in which neutral mutations in the first host caused by drifting genes that are useless in the actual host may be detrimental for the alternative new host. Trade-offs in the adaptation to different hosts are not always evident, and in our work where we analyzed the fitness of CMV isolates from different hosts, we did not find any significant differences in viral accumulation of isolates in the host of origin or in other hosts, which was actually not evidence of host adaptation. However, when we looked at infectivity, we did find differences in the bean isolates, which infected more bean plants than cucumber or tomato. After 10 passages in the host of origin, we found an increase in accumulation for bean and tomato isolates. This means that original fitness was not the maximum because it could still increase with the passages, and this was maybe due to the frequent changes of host during a virus cycle. Improvement of fitness in its own host did not entail a decrease in fitness in tomato and in bean, so there was no evidence of pleiotropy in this host, but passages in bean did show pleiotropy with accumulation in cucumber, and passages in cucumber showed a decrease in accumulation in bean and tomato, probably due to the accumulation of deleterious mutations for these hosts. So after this part, so we need to pay more attention to the question, what is the involvement of virulence factors in pathogen life stages other than infection? Also, experimental data about trade-offs in the evolution of virulence activities are often contradictory, and a little is known really about the molecular mechanisms that restrict these pathogen virulence in the cases where an observation in decreased virulence is observed due to these trade-offs. Speaking about the ability to infect different hosts, one of the most relevant consequences of novel virulence activities is, in fact, the ability to infect new hosts, which ultimately leads to the emergence of new diseases, as we have been observing during the last 2 years. The adaptation of pathogens to new hosts may lead to host range expansions, which is the adaptation to new host without direct influence on the gene pool of the pathogen, or host jumps, which is the genetic differentiation of a pathogen populations that are able to infect different hosts, and finally leading to pathogen speciation. The dominance of host range may be both intrinsic, which are the availability of virulence factors that allow the interaction with different hosts, and these factors can be subject to trade-offs and costs, and extrinsic, which is the exposure of plants to microorganisms and environmental conditions favorable to infection. So first, the pathogen need to have a repertoire that allows them to infect a new host, and also then it must have the opportunities to be in contact to that host to be able to infect. Also, if there are geographical and ecological isolations, this can lead to these host jumps by pathogen speciation. The first step in the ability to infect a new host should be overcoming the so-called non-host resistance. And MPMI top 10 question 6 is about its molecular basis and has been approached by Ralph Franstruga and Matthew Moscow. And you can see their very nice seminar that has been recorded in the MPMI seminar series. And since non-host resistance is mechanistically indistinguishable from other types of plant immunity, as they also say in this review, virulence factors for the adaptation to new hosts may not be very different from those deployed against host resistance. And a very nice example of this is this work of Inouye and collaborators about the host expansion of the fungus Pedicularia oryzae to wheat. Here is also remarkable the involvement of a Birch host. So isolates carrying PYT3, PWT3 virulence genes, probably coming from wild grasses, could adapt to wheat varieties lacking the resistant gene that recognizes this PYT3. And later, once these variants were able to infect wheat, new variants arose with mutated PWT3, and these were able to infect resistance with genotypes, which in fact are most of other varieties. And this variant became predominant in the population in Brazil in very few years, and expanded to Bangladesh, where also in very few years, where they are now causing severe crop losses. So, but also although I have brought here a very nice and clear example, this example of Magna Pedicularia orythae, of how can evolve the adaptation to new hosts. In fact, we still do not know most of the key steps that lead to the adaptation to new hosts and thus determine pathogen host range. Even molecular mechanisms associated with adaptive evolution of host-specific lineages remain mostly poorly understood. So I have spoken about some of the selective factors that affect evolution of novel virulence activities, but selection acts on genetic variation. And I would like now to mention some of the genetic mechanisms by which pathogens generate and recombine variants and the virulence activities that can result. We can divide these genetic mechanisms in mutation, which are changes intrinsic to each genome, and genetic exchange, which communicates changes between genomes. Within mutation, I would like to pay special attention to transposable elements and to the duplication and new functionalization of genes. About genetic exchange, we can see events occurring within species and kingdoms, or even between kingdoms. Mutation is defined as any heritable change in the genome of an organism that can occur at the level of one of a few nucleotides up to larger scales of genes or segments of chromosomes. We have already seen that mutations in virulence genes can give rise to pathogen genotypes able to overcome host resistance or acquire a new host. We have also seen that mutations can generate new variants able to interact with host factors. In several cases, it has been observed that some regions of the genome are more prone to mutations than others, generating genomes compartmentalized into gene-rich, slow-evolving regions and gene-sparse, repeat-rich, high-variable regions. These genomes are called two-speed genomes. In this work of Dong and collaborators, they describe this phenomenon in two very distant plant pathogens, a fungus, Leptosphaeria maculans, and an oomycete, Phytophthora infestans. Virulence genes in these two organisms were found in the gene-sparse, high-variable regions. Similar patterns have also been found in other fungi and in bacteria and nematodes. It is not clear if mutation rates are higher in these compartments, or rather, their evolution is the result of the absence of purifying selection. In fact, it is very difficult to know if there is a different mutation rate. Why? Because it is really complicated, and there are very few cases where mutation rates have been determined. Why this type of genomes? What is the advantage of this compartmentalization? Well, with the compartmentalization of virulence factors in these regions, allow the rapid evolution while minimizing the deleterious effects of mutation on the housekeeping genes in the other regions. So it keeps them apart, so these can evolve rapidly and these can be conserved. However, this pattern is not observed in all organisms. In the fungus Plumeria graminis, which is one of the largest fungal genomes, the putative secreted effectors are not found in a particular genomic region. And in viruses with small genomes with overlapping regions and multifunctional proteins, there is no room for high variable gene sparse regions, and mutations may have pleiotropic effects that lead to costs of virulence activities. This pattern observed in Plumeria graminis is probably related with the pattern of transposable elements in its genome, which are homogeneously interspersed along the whole genome. Meanwhile, in two-speed genomes, These are preferable localized in these particular regions of the genome that are poor in genes and rich in variability, and where many virulence factors are found. Indeed, these are powerful mutators in prokaryotic and eukaryotic genomes, and we have already seen that these were associated with variations with important consequences. And I have already shown AVRSTB6 and other variants in Septoria drittisi, and this gene is located in a genomic region completely surrounded by Ts. And in Pericularia oryzae, the pathogenic loss of function of PW33 variants are due to different Ts insertions. But the action of Ts is not just direct mutation by insertions in the affected gene. They also involve changes in surrounding genes due to genomic defense mechanisms such as epigenetic silencing or RIP mutations. These can also hijack and duplicate genes, and the repeats generated by their action increase the probability of recombinations. Even these can be sources of new virulence factors by their co-option and neofunctionalization. Here I show two very clear cases. Botrytis cinerea uses T-derived sRNAs to hijack host RNA interference pathways and suppress plant immunity. And here we can see that Botrytis strains transformed with a T are more virulent than wild type. And in barley powdery mildew, ROP-PIPE1, which is an effector peptide that promotes infection, clearly derives from a short interspersed TE element. Another thing, gene duplication and neofunctionalization, not just of TEs, is at the origin of many virulence factors. There are many examples of large gene families of effectors that have expanded in pathogen genomes and conserved signatures that reveal a single origin. And also the different processes of gene birth and death in different genomes produces the so-called pangenomes, with a core of conserved genes present in all individuals of a species and accessory different genes in different lineages. These accessory genes are associated with adaptive functions, among which virulence activities. One very clear example of an expanded and neofunctionalized family is the GS-like effectors of plant parasitic nematodes that were originated after two large-scale multiplication events of the housekeeping glutathione synthetase. This group of GS-like paralogs acquired canonical features of an effector, such as the expression in the dorsal gland and signal peptides. These bars represent the presence of signal peptides in this clade of paralogs. And these proteins, the encoded proteins, are translocated into host cells, forming the infection sites called syncytia, which is also typical of effectors. Another type of source of variability is the genetic exchange, sexual and parasexual. Genetic exchange can also give rise to these novel virulence activities. Recombination can generate novel combinations of virulence factors and purge deleterious alleles. So it has the potential to produce novel virulent phenotypes, sometimes more rapidly than mutation alone. For example, in virus, the contribution of recombination to the generation of genetic diversity can be comparable to that of mutation. And there are some types of viruses, such as, for example, DNA Gemini virus, where recombination events are very frequent. In this example, a natural recombinant between the two different species, tomato yellow leaf curl Sardinia virus and tomato yellow leaf curl virus, acquired the ability of infecting hosts of both parentals. One parental infected tomato and bean, and the other was able to infect tomato and wild solanums, and the recombinant was able to infect all these hosts. And this recombinant, though it is very little, maybe you cannot see, became prevalent in just 1 year after being detected for the first time. In eukaryotic organisms such as oomycete, fungi, and nematodes, intra and interspecific sexual hybridization events have been reported to evolve remarkable novel virulence activities, such as host range expansion in Phytophthora, Plumeria graminis, and nematodes, or the ability to overcome resistance genes as the UD99 variant of wheat stem rust. These are really remarkable gains of virulence activities, but the specific genetic mechanisms behind them should be something more than just recombinations and are largely unknown. Maybe they are related to the gain of genetic capital, but not only. It is not really very well known. Another question is the horizontal gene transfer. Horizontal gene transfer is the transference of genes or group of genes between lineages by means of parasexual mechanisms. In fungi, the signatures of horizontal gene transfer of virulence factors has been reported, and it is quite frequent the observation of the transfer of dispensable chromosomes that, although not essential for living, carry determinants of virulence activities. The mechanisms are not fully understood, but most probably are related with parasexual anastomosis events. For example, the horizontal transfer of partial chromosomes in Fusarium Oxysporum turns non-pathogenic strains in pathogenic. In bacteria, the horizontal transfer of integrated conjugative elements and plasmids can confer novel virulence capabilities to the receptor strains. For example, in these graphs showing the effector repertoire of recently emerged Pseudomonas strains infecting cucurbits, we can see the core effector repertoire of the pangenome and the different accessory effectors present in different strains. Different superscripts mean different sources, although they are very little. Maybe you can see the different superscripts, and some of them have 2 or more superscripts. That means that these effectors have been acquired through different sources among strains. Genomic analysis have also shown signatures of the horizontal gene transfer between kingdoms. I do not have time. I think I'm already out of time. I don't have time to detail them here, but there are several examples of virulence factors in nematodes, fungi, and oomycetes that have been ancestrally acquired from fungi, plants, or bacteria. So to finish, what are the unknowns about the genetic mechanisms behind the evolution of novel virulence activities? Well, there are still many, and some of them are, for example, that empirical estimates of mutation rate of most pathogens remain scarce and the underlying mechanisms unclear, that we don't really know very well which are the mechanisms underlying chromosomal variation and what it is specific impact on virulence, and what is the relevance of nature of parasexuality, and how often do genetic transfer events give rise to novel virulence activities occur in the field. Which are the underlying mechanisms of horizontal gene transfer between different species and kingdoms? What are the specific genetic mechanisms that increase virulence or host range of hybrids? And if novel virulence does more often evolve de novo or via the transfer of existing virulence factors from some other systems. And to finish, in summary, I think that we can conclude that if there is an epigenetic or genetic mechanism that can create variation in the genome, it will be used by pathogens. Classical loss of AVR seems the most common form of evolution of virulence, but this may be due to a bias in research rather than the reflection of reality. And there are many other underexplored virulence activities during the life stages of the pathogen. There are also many underexplored effects of actors other than the host. So, for example, biotic and abiotic factors in the ecosystem. I haven't have time to speak much about this. And more studies are needed to disentangle the involvement of virulence factors in pathogen life stages other than infection, and implications in coevolutionary processes and adaptation to new host. So I finish here. Many thanks for your attention. And if you have questions, I will be very happy to try to answer them. Thank you.
Thank you, Soledad, for a fascinating talk on a very big question. I love how you went into mechanisms as well as what's unknown. Why don't you stop sharing your screen?
Okay.
And then we'll be able to have everybody come up. Terrific. So I'm going to start with the first question from, and just to remind everybody, if you would like to ask a question of our speaker today, please type your question into the Q&A box and we will take turns reading them. And oh, also one other reminder that this has been recorded and you will be able to access the recording for free at the same place where you registered for this meeting. And so you can pass that on to people you know who missed it. Okay, first question from Mohammed Hook. Are vectors affected by the pathogens they carry? So is there any genetic change of the pathogen that happens during carrying by a specific vector? Very interesting question.
So yes, of course. I didn't include it very expressed, but there are works that show that the selection pressures for example, for viruses that infect both the plant and the vector, the selection pressures are different, and in some cases even they are contrary. So there are strains that are transmitted that multiply better in the vector and then worse in the host, and the opposite. So of course there are genetic changes. There are genetic changes during the transmission because the best fitted strains should be transmitted better, but also I haven't talked about it, but also because there are bottlenecks during the transmission, and these can also affect to the population structure.
Thank you. So we have a second question coming from Dave Berger, and he said that you started with a definition of virulence. Is the term aggressiveness an alternative term for virulence in plant pathogens, or does it mean something different?
Okay, so when Van der Planck used virulence in the sense of infectivity, actually, he then used the term aggressiveness in the sense of virulence. So you can use aggressiveness in the sense of virulence. In fact, well, the thing is that the term virulence, is used a bit different in plant pathology than in other disciplines. It should be better if we could use the same terms, but it doesn't matter if you always, when you are starting to speak about a term, you always define it and say, I'm referring to this. So then I think it is fine, the term you use.
Okay.
Okay, thanks for that. It's always so interesting how language frames the way we think about things.
Yes.
Our next question comes from Nanda Kumar. How do these important mutations miss the DNA repair mechanism?
Okay, yeah, well, mutations occur actually because other previous mechanisms that are there to avoid these mutations have failed. So sometimes it happens. So depending on the of the organism, it happens more often or or the repair mechanisms are better, and some organisms work less. So, for example, in viruses, RNA viruses, we know that there are no correction function of the polymerase. So yes, of course, DNA repair mechanism is very important to understand why the mutations are happening and how much do they happen. Yes.
Okay, thank you. Cynthia Perez asked whether the data are available, and I think she's asking for the reference, the review that you just presented on. So Cynthia, that issue is available on MPMI, and Ashley has posted a link in the chat. And this seminar, of course, is being recorded and it will be available to you online after we end today. So feel free to access the data either online, or you can look at the review and read it for yourself. So that was an easy question. So I think we can, let me ask you a scientific one now. And this one comes from Davis Zambrana. And he asked, in the long term, are genes under diversifying selection able to provide the most fitness advantage in the long term? So
I do not think that because they are subject to diversifying selection in the long term, they are going to, I don't know, they are going to bring more fitness advantages. I really do not know. I don't know if we can forecast the fitness in the long term, thinking about the selection, the type of selection. The type of selection is telling us about the process that they have followed until now, but we cannot know what is going to happen in the future. Yeah. We can just know if a population has a lot of diversity, that it has a higher potential to evolve. This is what we can know. Yes.
Yes, exactly. Okay, we have another question. Oh, before we get to that, I just wanted to say that in addition to being recorded, we will have a caption so that people who are hard of hearing or for whom English is not their first language can follow this on closed captioning. And while Zoom provides an automatic transcript, as all of you know, for scientific language, you need a scientific reader to update and correct. So if anybody would like to volunteer for this important job to help make our seminars more available, more inclusive to our wider community, please type your name into the chat and we will be in touch with you. People have very kindly volunteered their time for this in the past, and we really appreciate this effort to make our seminars more accessible. Okay. While you guys are thinking and typing into the chat, I'm going to read our next question, which comes from Aravind Kumar Konda, who is wondering about a chitin binding protein from Magnaportha, which is used as an effector protein that silences PTI, whereas there's I think it's a tetratricopeptide protein from rice, which prevents the inhibition of PTI. And so he's wondering about a chickpea protein he has found, a chickpea gene, which is a chitinase with a tricopeptide domain. What are your views on this? So this is sort of a, I guess, a fusion. If you take two genes that influence the immune system and they have recombined into one gene, of course, this is exactly the kind of processes and events that Soledad has been talking about. So what do you think, in your opinion, might happen?
Well, I'm not really expert on this, but it looks like it could be an interesting resistant genes in chickpea. The question is knowing against which pathogen. So, well, it is quite interesting because it is having a function. If it is having a function as chitinase to make free the monomers of sheeting, and then also to the C peptide to stop the possible effector function of the pathogen to interfere with the detection. Maybe it could be an interesting gene to explore as resistance genes. Yes, could be.
Great. That was a hard one. So here's one I think that you could probably answer easier. If a particular genotype of a pathogen is more common in a population, can we suppose that this variant is under positive purifying selection?
Of negative purifying selection.
Yes, yes. If the genotype is most common within a population.
So well, we will have to see. Yes, it could be. If this pathogen is more common in the population, it is probably that it has been selected. It's under selection for increase its frequency because it is more fitted. So this is what the theory says, but not really is the direct. So more things should be looked at because it can also be subject to stochastical processes. So this is not just the only, the definitive clue. It is a very important clue, but not definitive.
Okay, our next question is fairly specific. It's asking, if you were to study Fusarium oxysporum, how would you study virulence changes? Like, where would, I guess the question is really, what would be your starting point? Because you've addressed so many different ways that virulence can change.
Mm-hmm.
What do you think would be a good starting point?
Well, I think with vascular I am not expert on vascular pathogens, but the thing is that I'm not sure if always virulence is completely linked. So virulence in the sense of harm to the host is always completely linked to the fitness of the pathogen, so the multiplication. So I would always look at the same time to the symptoms, but also at the same time to the ability of the pathogen to multiply. You know, there are several ways to do that. You can just see it by isolating, or you can develop molecular methods to analyze. So I would always use both measurements to be sure that there is a harm in the plant related to the ability of the pathogen to multiply within the plant.
Excellent. So before we come to the last question, I want to draw your attention to a post just placed in the chat by Morgan Carter. She's one of our associate features editors, and she is actually advertising that MPMI is currently looking for new associate featured editors, and she's provided a nice link and description of the sort of task that these associate features editors do. So please, you can follow up in the chat, the link that Morgan has placed. And so, Soledad, I think we have a probing question that everybody in this group probably wants to know. Can a pathogen lose its virulence upon repeated subculture in the lab? I think that's a long debate we've had in the field, right? About keeping our pathogens alive in the lab. So
yeah, that's a fear we always have, and I guess it can. I haven't had this problem yet, I think, but maybe yes. I am having it, but I don't really know, because it can be losing little by little. It can be accumulating mutations that. So as I have shown by changing hosts, it can also happen that when you are maintaining it in an artificial medium, that it is accumulating mutations that then make it less fitted for the infection of hosts. it is plausible that happens. What I had in my experience of serial passages is that also you have bottlenecks, and you can also, I lost infectivity in virus strains or virus isolates because I was doing without wanting it bottlenecks, and this is really very disruptive for clone populations that are accumulating mutations, and they cannot recombine to eliminate them from the population. So I think, yes, it is possible. It is recommendable to inoculate them in the plant sometimes.
Yes.
Yeah, and just to add to that, there's a classic example from symbiosis. Rhizobium bacteria can, of course, grow very nicely on a Petri plate, just like in the plant. But after, this was long ago, people had repeat passages. And of course, things that make a bacterium better for the scientist to grow on a Petri plate are things like less goopy, so you can pick single colonies better. And so, of course, the lab strains have vastly decreased exopolysaccharide production. And we know that exopolysaccharides are important for infection. So that's a beautiful example of the scientist inadvertently applying a selective pressure by saying, oh, look, I can finally pick a nice, clean single colony. I'll grow up everything from that. So just remembering how much we influence our subjects, that's a great question to end on.
Yes.
So this has been a lot of fun, and thank you very much, Soledad, for for this wonderful and very interesting seminar. I just want to remind everybody that this paper is available in MPMI. It's open access. So if you want to get all the details and all the references, things you might have missed in the talk, that's a great way to go. And you can also find Soledad's contact information in there, just as we always have for our corresponding authors. And finally, if you missed it, or if you know somebody who missed it, because I know plenty of people here who are watching from around the world, and so for some of you it's midnight or 3 a.m. or something. So and probably many of your colleagues are happily sleeping. So please let them know about the free recording. And once we get a corrected transcript, we'll upload that. And then it will be easier, I think, for people to follow. And of course, if you missed, you can also follow Morgan Carter, our assistant features editor, was live tweeting the whole seminar. And so all of that's available. You can follow step by step the seminar by looking at the MPMI Journal Twitter account. And that gives you, if you're interested in becoming one of our assistant features editors, that gives you an idea of one of the fun things you can do is periodically take over MPMI's social media accounts and connect with the whole community across the world. Okay, well, thanks everyone for joining us. Please thank me in thanking Soledad Sacristán for an excellent talk. And I hope you will all join us in 2 weeks for our next seminar by Sonali Roy. Bye, everyone.
Bye.